The extrinsic pathway of coagulation, also known as the tissue factor pathway, is one of the two pathways that eventually leads to the formation of a blood clot to stop bleeding. These pathways and all their clotting factors can be quite confusing, but not to worry. After watching this short video, you'll have a foolproof way to remember everything you need to know about the extrinsic pathway. Let's get started. We're at an outdoors picnic, and what better summery snack is there than red Jell-O? Yep, take a look at the to-go cups of Jell-O we're eating in this outdoor setting. By the way, the outdoor setting should remind you of the extrinsic pathway. You know, since extrinsic refers to things coming from outside, and the red Jell-O is our recurring symbol for coagulation, since Jell-O is formed by the solidification of red liquid, which is kind of like how red blood clots in our body, right? Putting this together, the outside Jell-O is our memory anchor for the extrinsic pathway for coagulation. For context, the extrinsic pathway is one of two starting pathways for making a stable blood clot to stop bleeding, a process also known as secondary hemostasis, since the formation of a blood clot occurs after the formation of a platelet plug, which is otherwise known as primary hemostasis. Focusing on the blood clot formation, there are actually two distinct pathways to start the formation of a blood clot. These two different initiating pathways are known as the extrinsic and the intrinsic pathways of coagulation. Both of these two pathways later converge into a common set of ending steps, known as the common pathway of coagulation. For this video, we're going to focus only on the extrinsic pathway, named since it depends on factors originating from outside the blood vessel. Kind of like how we're outside here, right? Now, when you sat down outside to enjoy your Jell-O, you just had to be a klutz and knock over your bottle of 7-Up, since nothing's better for a hot outdoor day than a cold bottle of 7-Up. Ugh, now this 7-Up has been spilled everywhere. Coincidentally, this 7-Up is here to help you remember Factor 7. This one's pretty straightforward, 7-Up for Factor 7. Factor 7 is a circulating protein in our blood that kicks off the extrinsic pathway when it comes in contact with something from outside our blood vessels. This contact causes the cleavage or splitting apart of Factor 7, which actually activates the protein. As for what reacts with Factor 7 to activate it, let's talk about that next. To clean up your spill of 7-Up, you're reaching for the closest thing, a box of tissues. Of course you have tissues here in this outdoor picnic since allergies get pretty bad outside, right? Anyways, this box of tissues should remind you of the tissue factor, the thing that reacts with Factor 7 to kick off the extrinsic pathway of coagulation. Tissues for tissue factor. Get it? Tissue factor is normally expressed by cells that are not directly exposed to flowing blood, such as the subendothelial smooth muscle or fibroblasts surrounding the outside of blood vessels. In other words, tissue factor is normally found in tissues outside the reach of flowing blood, and its presence inside the blood is abnormal. So when tissue factor reacts with Factor 7 inside the blood, this is really just a fancy way of saying that tissues from outside the vessel have been exposed to blood. This means that an injury has happened to the blood vessel wall and that there is a need to start the blood clotting pathway. This is why the tissue factor pathway is also called the extrinsic pathway of coagulation, since it requires tissue factor which originates from tissue outside the path of flowing blood. Next, check out what you also brought to cool off on this hot summery day. Yep, that's a cup of calcium ice cream. I think you were going to mix the 7-Up with this ice cream to make an ice cream float. Anyways, you probably know that this calcium ice cream is our recurring symbol for calcium. And much like you were going to mix up the 7-Up with this calcium ice cream, Factor 7 requires the presence of calcium ions for activation. The rationale here is complicated, but in a nutshell, the charged calcium ions change the conformation of Factor 7 to catalyze its binding to tissue factor. There's actually a few more catalysts here that are needed. Let's talk about these next. Next, let's check out the membrane seal that has been half peeled away from the ice cream. You know how these to-go ice cream cups always have this membrane seal you have to peel away? Well, the membrane here kind of reminds me of phospholipids, since phospholipids are the things that actually make up the membranes of our body, right? This is here to help you remember that phospholipids, or the presence of a cell membrane, is also important for the activation of Factor 7. There's one more ingredient to add to this mix. Let's get to that next. When making an ice cream float, there's nothing better to add texture and some crunch than some Super K cereal. Wait, Super K? Isn't that Pixarize's recurring symbol for vitamin K? Yep, this Super K cereal is here to help you remember vitamin K, another required component for activation of Factor 7 in this pathway. In fact, Factor 7 is one of the vitamin K-dependent clotting factors. Clinically, this is relevant because Factor 7 and the extrinsic pathway is actually the primary target of warfarin, a blood thinner drug that works by preventing the activation of vitamin K-dependent clotting factors. Just know that vitamin K is needed for the activation of Factor 7 in the extrinsic pathway, okay? So what does this activated or cleaved part of factor 7 actually do? Well, take a look right at your friend with the tennis racket. Yep, after your picnic here, you were going to hit the courts next to work on your swing. And you only practice with the best tennis equipment, as evidenced by that 10 out of 10 rating on the tennis racket. By the way, the tennis racket and the 10 out of 10 rating should both remind you of factor 10. Got that? Activated factor 7 next works to cleave factor 10, activating it in turn to cleave other things. The activation of factor 10 is actually the end of the extrinsic pathway of coagulation and the beginning of the common pathway of coagulation, which eventually leads to the formation of a stable blood clot. Check out our next video on the common pathway to learn more. Before we close, let's touch on how we can monitor the efficacy of the extrinsic pathway. Take a look at the paper towels your friend is holding out to help you clean up your spill. The paper towels here are a recurring symbol for PT. You know, if toilet paper is TP, then paper towels have to be PT, right? PT actually stands for prothrombin time, a laboratory measure for how well the extrinsic pathway is working to clot blood. Things that block the extrinsic pathway, such as the drug warfarin, will lengthen the amount of time it takes to form a blood clot in what is measured as the prothrombin time. The takeaway here is to recognize that PT is used to measure the extrinsic pathway. Clinically, you'll want to distinguish PT with 1 T from another test, the PTT with 2 Ts, since PTT, or partial thromboplastin time, is a measure of the intrinsic pathway, a totally different pathway than this one here. Just remember the paper towels were needed to clean up your 7-Up mess outside, and you'll never forget that PT measures the extrinsic pathway. Alright, that's it for the extrinsic pathway of coagulation. Let's summarize. The extrinsic pathway starts when factor 7 circulating inside the blood is exposed to tissue factor from outside the blood, signifying an injury to the vessel. This reaction causes the cleavage and activation of factor 7 in a process that also requires calcium ions, phospholipids from a membrane, and vitamin K. The activated factor 7 then goes on to cleave factor 10, activating that protein to go on to cleave other things. The activation of factor 10 is the end of the extrinsic pathway and the beginning of the common pathway of coagulation. Importantly, we can use the PT, or prothrombin time, to monitor the status of the extrinsic pathway of coagulation. Phew! We're all done here. Get yourself a 7-Up and tell your friends if you enjoyed this video. See you next time!